All right, let's just jump right into it. Today, we're diving into this fascinating explainer all about the incredible journey of how humanity actually fights back against our microscopic enemies. I mean, for centuries, we were literally losing the battle against these invisible forces. But today, we're right on the brink of predicting and stopping them before they even emerge. This right here is the story of virus hunting. To truly grasp where we're headed in a post COVID-19 world, we're going to walk through a roadmap of 5 key eras. We'll start with the dawn of virus hunting, move into the golden age of vaccines, hit the genomic revolution, cover global surveillance and zoonosis, and finally wrap up with AI and the future. Let's get to it. Before we really get into the timeline, I want to anchor us with this amazing quote from the 1960 Nobel laureate Peter Medawar. He said, "A virus is a piece of bad news wrapped in protein." And honestly, this just brilliantly captures the fundamental problem humanity faced. We're talking about an existential threat wrapped in this incredibly simple yet devastatingly efficient little package. See, a virus lacks the cellular machinery to replicate on its own, right? So what does it do? It hijacks ours. It truly is, literally, just a piece of bad news wrapped up in protein. So let's start with section 1, the dawn of virus hunting. This was a wild time, honestly, when scientists were fighting an enemy they couldn't even see. Looking at this historical timeline, it's just amazing to realize that back in the late 19th century, early pioneers like Adolf Meyer and Dmitry Ivanovsky deduced the existence of viruses through, well, pure deduction. They used these special porcelain filters that were actually designed to trap bacteria. But when the liquid that passed through the filter still remained contagious, wow, that's when they knew an invisible, unimaginably tiny pathogen was at work. This kind of indirect experimental observation really kicked off a methodological cascade that completely defined the dawn of virology. Of course, as our ability to hunt these filterable agents grew, we needed a solid way to prove that a specific virus actually caused a specific disease. So, scientists developed this rigorous hierarchy of confidence. This was established by researcher W. Ian Lipkin. It basically moves in stages. You start with a possible relationship, like finding viral nucleic acid in a patient. Then it moves to probable, or you have a biologically plausible link, or maybe a super high concentration of the virus right at the pathology site. And finally, we hit confirmed. This means fulfilling Koch's postulates. Essentially, you're reproducing the disease in a host, or better yet, preventing it entirely with a vaccine. All right, moving on to section 2, the golden age of vaccines. Because once we understood the cause, well, we needed weapons. These early efforts laid an absolutely incredible foundation for eradicating some of humanity's oldest scourges. Just take Edward Jenner's 1796 cowpox inoculations against smallpox, for example. Jenner essentially pioneered the whole concept of vaccination. And then, decades later, you have Louis Pasteur introducing attenuated vaccines for rabies. These foundational breakthroughs were a massive game changer. They allowed us to move from just passively observing these devastating diseases to actively training the human immune system to fight them off. And as you can see, the compounding nature of technology has drastically compressed the time it takes to develop these life-saving interventions. Think about it. It took nearly a century to get from the smallpox vaccine to the rabies vaccine, 100 years. But as cell culture techniques evolved by the mid-20th century, boom, we saw the polio vaccine emerge. And then, fast forward to 2020. By leveraging decades of whole genome sequencing and recombinant techniques, safe and highly effective COVID-19 vaccines were developed and deployed globally in literally a matter of months. That's just wild. Which perfectly brings us to section 3. The genomic revolution. So, for decades, clinical virology steadfastly relied on traditional diagnostic approaches, things like viral culture and serology. Now, don't get me wrong, traditional culture methods definitely remain vital, especially when we're testing drugs and vaccines, but they are inherently slow. They often require extensive time just for incubation. Today, though, molecular assays like rapid PCR have pretty much superseded those older methods to become the gold standard. They provide this exquisite sensitivity and rapid speed that is just absolutely required for clinical decision-making. And as we all know, that is absolutely critical during a pandemic. But the real game changer here has been high-throughput sequencing, or, as it's also known, next-generation sequencing. Instead of using targeted amplification, which, only looks for viruses we already know about, this technology extracts sequences directly from complex environmental or clinical mixtures. This essentially freed virologists from relying on predefined genetic markers. It means we can now detect massive numbers of uncultivated, completely novel viruses. And the economics behind this are just staggering. To put it into perspective, the cost plummeted 10,000-fold in just a decade. We went from paying $5,000 per thousand genetic letters, or nucleotides, back in 2001 to just 50 cents by 2012. 50 cents. That is unbelievable. This incredibly data-heavy approach allows us to catalog vast amounts of genetic material into global databases, like GSIDE. It basically gives us a real-time global view of molecular epidemiology. So, for instance, sequencing efforts during outbreaks allow public health officials to identify new variants, track transmission routes, and spot drug resistance mutations almost immediately. The big takeaway here, we are no longer just reacting blindly. We are analyzing the viral blueprint in real time as it evolves. Moving on to Section 4, Global Surveillance and Zoonosis. Because sequencing is really only half the battle, right? We also have to monitor where these threats are actually coming from. And here's the thing, the vast majority of our emerging crises, we're talking HIV, SARS, Ebola, they're all zoonotic, meaning they jump the species barrier from wildlife or domestic animals directly into humans. And what's driving this? Well, it's a mix of human incursions into wildlife habitats, consumption of bushmeat, super high population densities, and of course, a warming climate that's expanding the range of insect vectors. It is the ultimate One Health challenge. Human, animal, and environmental health are just totally, completely interconnected. To really illustrate the massive scale of this vulnerability, consider this absolutely astonishing number. An estimated 90 million tons of bushmeat are trafficked annually through JFK Airport in New York alone. 90 million tons. Analyses of seized wildlife products have actually revealed hidden retroviruses and herpes viruses being carried right into densely populated, highly connected urban areas. I mean, in an era where nonstop flights connect the world's major airports in under 24 hours, an isolated animal virus can literally become a global human crisis overnight. Now, okay, I know this might sound a bit terrifying, but there is actually incredible hope in the data. Researchers recently studied the Indian flying fox, which is a known viral host, to kind of extrapolate the total number of undiscovered mammalian viruses out there. And they estimate there are around 320,000 awaiting discovery. But here's the cool part. Economic models applied to the status show something really remarkable. We can proactively identify 85% of this vast viral diversity for just a fraction of the total cost. Sure, finding that last 15% yields diminishing returns, but discovering the vast majority of these threats, that is highly achievable through targeted global biosurveillance. And that brings us to our final stop, Section 5, AI and the future. Because as our datasets grow exponentially, human analysis alone just simply isn't going to cut it. This is where AI swoops in and completely supersedes our old computational limits. See, traditional detection relies on comparing new samples to existing databases. But if a virus is completely novel, well, it just gets missed. Sequence-based AI, however, uses deep learning to look way past those exact genetic matches. Instead, it recognizes the overall mathematical shape and the hidden patterns of a virus. This allows it to detect previously uncharacterized pathogens, stuff scientists refer to as viral dark matter. How cool is that? By combining machine learning with advanced proteomics, which is just the study of viral proteins, we can actually identify exactly how a virus enters a cell and from there find novel drug targets. Meanwhile, systems biology steps in to let us map out the entire host response, integrating cellular RNA and metabolic processes all at once. The AI essentially ensures we aren't just staring blindly at massive noisy datasets, but that we're actively finding the actionable, life-saving patterns hidden within them. This incredible synthesis of technology and biology means our defensive toolkit is completely transforming. We're transitioning away from purely reactive treatments and moving toward highly proactive, data-driven suppression. By understanding the local sequence and fitness landscape of a virus in real time, our ultimate objective is clear. We want to integrate these computational tools to literally push deadly pathogens toward evolutionary dead ends. Essentially, we want to trap them and drive them straight to extinction. Considering how far we've come, from literally filtering invisible agents through porcelain in the 1890s to deploying AI-powered deep learning against viral dark matter today, the trajectory is pretty clear. The power to stop the next global outbreak before it even begins, it might already be hidden right there in our data. The real question is, are we ready to look closely enough to find it? Think about that for a second. And thank you so much for joining me in this explainer.